This method provides solutions for today's demanding biological questions. From an in-cell FPOP experiment, we can study protein-ligand interactions, protein-protein interaction sites, and regions of conformational change. The advantage of this technology is the use of an automated 6-well plate-based IC-FPOP platform that makes it possible to grow cells and perform in-cell FPOP studies right at the laser. This method can be used in pharmaceutical and biotech industries, as well as major academic and clinical research institutes, especially by biological investigators and universities studying protein dynamics.
To begin, unscrew the platform incubator from the positioning stage and disconnect the temperature, gas, and humidifier lines. Spray the incubator with 70% ethanol and place it in the cell culture hood. Remove the 6-well plate from the platform incubator, secure the plate's original lid, and confirm cell confluency using a microscope. Then place the 6-well plate with confluent cells back in the platform incubator inside the cell culture hood. Insert 3 pre-cut Tygon tubes in each well via the embedded ports by flushing the tubes to the well walls and secure them with custom 3D printed rings. Prepare an integration software script for pump withdrawal. Use one peristaltic pump to completely remove cell media from all 6 wells. Prime solvents in each channel of the other 3 peristaltic pumps. Then infuse hydrogen peroxide and quench buffer in their respective alternating tubes until the reagents reach the incubator ports. Confirm that the laser beam has been angled correctly.